Hi everyone, sorry about that. So today I'd like to talk to you about the potential of mushroom and vitamin D2 enriched mushroom powder in weaned pig diets. So weaning is one of the most stressful and challenging phases of a pig's life. So in the wild, piglets are gradually weaned at around 10 to 12 weeks of age. However, on commercial pig farms, to increase productivity, we wean pigs at 28 days of age, at a time when the piglet's digestive and immune system is still immature. And this can cause a cascade of events to occur that can impact pig growth performance. So at weaning, pigs face multiple stressors as they're moved away from their mother and mixed with piglets from different litters, and they're moved from a highly digestible milk-based diet to a less digestible cereal-based diet. So as a result of this, we often see a reduced feed intake in the immediate post-weaning period, and it can take anywhere between 7 to 14 days for piglets to reach the pre-weaning feed intake levels. So due to this reduction of feed intake, we can often see damaged intestinal architecture. And if we look over here at the image on the right-hand side, this is a section of a healthy gut. And here we can see long finger-like projections called villi. And the larger these are, the better, as it means there's more surface area for nutrient absorption. However, post-weaning, these villi become short and stunted, meaning nutrients can pass undigested and unabsorbed through the small intestine and into the large intestine, where they stimulate the growth, where they act as a substrate for the growth of pathogenic bacteria. As well as this, during the post-weaning period, we see reduced gut barrier function, and this allows pathogenic bacteria and antigens to cross the epithelial barrier, and this enhances immune system activation and increases the production of pro-inflammatory cytokines. This cascade of events that occurs ultimately leads to increased incidences of post-weaning diarrhea and reduced growth performance in pigs during the post-weaning period. So farmers use zinc oxide and in-feed medication in the post-weaning pig diets to prevent the post-weaning growth check from occurring. However, the EU are banning the use of zinc oxide and in-feed medication in 2022 due to their role in antimicrobial resistance. In light of this, there's urgent need to find suitable alternatives that can replace zinc oxide and in-feed medication and prevent this cascade of events from occurring during the post-weaning period. So some bioactive compounds that may beneficially affect the health of the weaned pigs includes beta-glucans and vitamin D. So beta-glucans are a type of polysaccharide found in fungi, cereal grains, and seaweed. And beta-glucans derived from fungi and yeast are connected with beta-1,3 and 1,6 linkages, and they demonstrate anti-inflammatory, antioxidant, and immunomodulatory properties. And as well as this, beta-glucans can positively influence the gut microbiota to increase beneficial bacteria populations, such as lactobacillus and bifidobacteria. So as well as this, vitamin D also has the potential to inhibit the cascade of events that occur during the post-weaning period, as it plays an important role in the modulation of inflammation by increasing the production of antimicrobial peptides and anti-inflammatory cytokines, and reducing the production of pro-inflammatory cytokines. And as well as this, vitamin D plays an important role in maintaining a healthy epithelial barrier and gut microbiota through the reduction of pathogenic bacterial species, such as E. coli. And mushrooms offer a unique way of incorporating both beta-glucans and vitamin D into the diet of the pig. So mushrooms contain an array of bioactive compounds, such as phenolic compounds, vitamins, and beta-glucans. And they're also a natural source of vitamin D, as they contain high levels of ergosterol. And ergosterol, when exposed to UVB light in a controlled process post-harvest, mushrooms can convert, ergosterol can be converted to ergocalciferol, also known as vitamin D2, making mushrooms a great source of vitamin D. So with this in mind, the objective of my experiment is to compare the in vivo efficacy of mushroom powder and vitamin D2 enriched mushroom powder with zinc oxide on piglet gastrointestinal health during the post-weaning period. And the hypothesis is that the supplementation of mushroom powder, in particular vitamin D2 enriched mushroom powder, would enhance gastrointestinal health similar to zinc oxide by improving intestinal morphology, reducing inflammation, and altering microbial populations during the post-weaning period. So for my experiment, we had 192 weaned pigs with an average weight of 7.8 kilograms, and they were randomly assigned to one of 4 treatment groups. The first treatment group was a basal diet, which was a control group. The second treatment group was a basal diet supplemented with mushroom powder. The third treatment group was a basal diet supplemented with vitamin D2 enriched mushroom powder. And the fourth treatment group, which was a positive control, was a basal diet supplemented with zinc oxide. So all the diets contained a vitamin D3 content of 50 micrograms per kilogram of feed. And then the vitamin D2 mushroom powder diet contained an additional vitamin D2 content of 100 micrograms per kilogram of feed. And all mushroom diets contained 200 parts per million of beta-glucan. The pigs in feed were then weighed weekly for average daily gain, average daily feed intake, and food conversion ratio. And food conversion ratio is a measure of how efficient a pig is. So after a period of 42 days of supplementation, 1 pig per pen was humanely sacrificed for sample collection. And we collected tissue from the duodenum, the jejunum, and the ileum for histology to measure villus height and crypt depth. And we collected tissue from the duodenum, the jejunum, and the ileum to analyze the expression of genes involved in nutrient transport, immune function, barrier function, and short-chain fatty acid transporters. And then we collected digesta from the cecum to measure volatile fatty acids and to measure the makeup of the microbiome. So now I'm going to bring you to my results section. So on the y-axis is a variable measured, and on the x-axis is the treatment groups. So in blue, we have the control pigs. In yellow, we have the mushroom supplemented pigs. In the gray, we have the vitamin D mushroom supplemented pigs. And in the red, we have the zinc oxide supplemented pigs. So here we have average daily feed intake measured in kilograms per day. And as we can see here, the mushroom supplementation and vitamin D2 mushroom supplementation significantly reduced average daily feed intake in pigs compared to both the zinc oxide group and the control group. And as feed intake is the biggest driver of growth, we see a significant reduction in average daily gain in the vitamin D mushroom supplemented pigs and mushroom supplemented pigs in comparison to the zinc oxide group. Although we saw a negative effect on growth performance, we saw positive results in the small intestinal morphology. So here, looking at villus height in the duodenum, mushroom supplemented pigs, vitamin D mushroom supplemented pigs, and zinc oxide supplemented pigs all had a significantly higher villus height in the duodenum compared to the control group. And then if we look at the ileum, the villus height in the ileum, vitamin D mushroom supplementation and zinc oxide supplementation significantly increased villus height in the ileum compared to the control groups. That's a really positive finding. So now moving on to gene expression in the small intestine. So firstly, we're looking at the pro-inflammatory and anti-inflammatory cytokines. So interleukin-8, which is a pro-inflammatory cytokine, this was significantly reduced in the vitamin D mushroom supplemented pigs in comparison to the zinc oxide supplemented pigs. And then moving on to interleukin-6, which is also a pro-inflammatory cytokine, this was significantly reduced in the vitamin D mushroom supplemented pigs and the zinc oxide supplemented pigs in comparison to the control groups. And then IL-10, interleukin-10, which is an anti-inflammatory cytokine, this was significantly increased in the vitamin D mushroom supplemented pigs in comparison to both the control and zinc oxide groups. And now if we look at the nutrient transporter and the vitamin D receptor, so the peptide transporter 1 was significantly increased in all 3 treatment groups in comparison to the control group. Fatty acid binding protein 2 was significantly increased in all 3 treatment groups again compared to the control group. And then vitamin D receptor was significantly increased in the vitamin D mushroom supplemented pigs in comparison to both the control and zinc oxide supplemented group. So now if we look at the microbiome analysis at phylum level, the sequences were analysed using the open source software package Quantitative Insights into Microbial Ecology. And the dominant gut microbial phyla here were Firmicutes and Bacteroidetes, making up over 90% of the gut microbiota. However, there was no difference in the abundance of bacteria at phylum, family, or genus level between the treatment groups. So to conclude, dietary supplementation with vitamin D2 enriched mushroom powder improved intestinal morphology, increased the expression of anti-inflammatory cytokines and nutrient transporters, and reduced the expression of pro-inflammatory cytokines in the small intestine. And as the pig is a good model for human health, vitamin D2 enriched mushroom powder may play an interesting role in inflammation-associated diseases due to its immunomodulatory potential and ability to resolve inflammation. And the mechanism of reduced feed intake needs further exploration. Thanks for your attention, and thanks for giving me the opportunity again to present my work today.